Friends, today I am presenting the gate control theory of pain. It's a brief version of the gate control theory. Here is a picture I have made. I would explain in the coming slides. Before we try to go to the gate control theory, let us understand what is pain and the pain pathways. Pain is an unpleasant sensation with actual or potential tissue injury or damage. The receptors for the pain are known as nociceptors. They are free nerve endings. These are the free nerve endings. These these free nerve endings are carried by two type of fibers: the thinly myelinated A delta fibers and unmyelinated C fibers. So they are carried by a dual pathway, A delta and C fibers. These fibers reach the spinal cord. In the spinal gray matter, C fibers are relayed in 1 and 2, and A delta fibers are relayed in 1, 2, and 5 laminae. From the fifth laminae, the wide dynamic range neurons originate, or on the other hand, they are also known as projecting neurons. These projecting neurons, they send axons to the thalamus. So these wide dynamic range neurons, they are relayed and they receive input from layer 2 and layer 1, that is from the substantia gelatinosa cells. The axons of these projecting neurons are the WDR. They cross to the opposite side and ascend as lateral spinothalamic tract. There are at least 4 different pathways associated with the lateral spinothalamic tract. The first one is neospinothalamic tract, or that is the lateral spinothalamic tract for carrying the neo sensations, that is the discriminatory sensations, that is about the attributes that will be relayed into the specific nuclei of the thalamus, that is the ventroposterolateral nucleus of the thalamus, where it joins the medial lemniscal sensory relays, that is the fine touch and all those things. Then they are relayed to the post-central gyrus, the Brodmann area, that is for understanding the location and the type of pain. Then we have another pathway that is known as a paleospinothalamic tract. This paleospinothalamic tract, thalamic nuclei involved here are dorsomedian nucleus and the ventromedian posterior nucleus of thalamus. These two nuclei are involved, and this is known as a paleospinothalamic tract. And they are also going through the reticular system and the parabrachial nucleus. This is parabrachial nucleus. Or the dorsomedial nucleus of thalamus projecting to the anterior cingulate cortex, anterior cingulate cortex. The ventromedian posterior nucleus of thalamus, that is projected to insular cortex. In addition, the parabrachial nucleus projects to amygdala. Amygdala in turn project to the anterior cingulate cortex and the insular cortex. Okay, this is about the projection. The purpose of these fibers is the emotional component of the pain or affective component of the pain. The neospinothalamic tract is for a discriminatory component, that is epicretic. Affective component, this is protopathic, and it will try to know what is that pain and what are the reactions of the pain. Then we have another path or end up the in the reticular system. These are spinoreticular pathways. And From the reticular system, once the reticular system, there are projections to thalamus. These are reticulothalamic projections. From the spino-reticular system, that means reticular system starting from the medulla to the pons and the midbrain. Then from the reticular system that would reach to thalamus, the non-specific nuclei that is in the internal medullary laminae of the thalamus and the reticular nucleus of the thalamus. And from these thalamic nuclei, non-specific thalamic nuclei, it is relayed into the entire cortical area, that pan-cortical areas, entire cortical mental. That is for arousal response. Fourth thing is, there is a projection to the periaqueductal gray. This is periaqueductal gray in the midbrain. And these are known as spino-mesenchymal fibers. And these periaqueductal gray and spinomedullary, that is reticular, spinoreticular, that is relayed into the rostroventral medullary area. These two things give a descending inputs to the lamina 5 or lamina 2 neurons, lamina 5 or lamina 2 neurons for controlling the pain sensation, how that is transmitted. So that means the controlling system is spino-mesenchymal, that activates the descending inputs, they will come back. And this is the descending input. They will either allow or disallow the movement. Now, yes, you guessed right. It is the toll gate for the movement of the vehicles, especially the car. Here is the automatic toll gate that will the signal is sensed, and after sensing the signal, it reads and then it opens the gate. Suppose if the validity is not there, it will not open the gate and he will be penalized 2 times or whatever. So that type of the automatic gate system is available for the motor vehicle. Similarly, in the pain transmission, we have the toll gates. And these toll gates work for the transmission of the impulse from the spinal cord to the brain. These from the spinal cord to the brain, when they cross to the opposite side, especially that which one is the wide dynamic range neuron. Now let us see these pain fibers. They are small diameter fibers, either A delta fiber or C fiber. They are smaller diameter. When they come here, they would first reach the target cell, that is a WDR, or the projecting neuron, that would send impulses to the brain for the epicretic component of the pain. That's understanding the location and the attributes of the pain. Then at the same time, it would have a synaptic contact with the substantia gelatinosa cells in layer 1 and 2 of the lamina. And when what these do, they would inhibit the substantia gelatinosa cells. What happens if the substantia gelatinosa cells are inhibited? What happens to this transmission? As such, the substantia gelatinosa cells sends inhibitory impulses to the target neurons in this synapse. There is the inhibitory transmission. Now, this inhibitory transmission is inhibited. This is called disinhibition. It disinhibits the substantia gelatinosa activities. Once it is inhibited, what happens? It will allow the transmission of the impulses. It will not close the gate. It will allow the transmission of the impulses. The pain continue to reach to the brain. And we have another set of fibers, that is a touch fibers, A beta fibers. These are not for the nociceptive sensation. And when they are also activated whenever there is some these things, they are also activated or they can be activated by some means. When they are activated, what they do, they would send their normal response pattern and they will activate the target cells. Okay, they will activate the target cells to transmit the impulse. Okay, now the main purpose of this thing is they will also activate the substantia gelatinosa cells. If the substantia gelatinosa cell is activated, it would inhibit the transmission of the small diameter fibers or a pain sensation. So this is a closure of the gate. So that means the large diameter fibers close the gate. The small diameter fiber, that is stimulation, that opens the gate, that transmits the impulses to the brain. Okay, so now this is two aspect. The third aspect, from the brain, there is that is sub midbrain or the medullary level. There are descending projections to these target cells as well as to the substance. In brief about the gate control proposed by the Melzack and Wall. Now, this gate control is of importance for relieving the pain uh, in whenever you are in acupuncture. So acupuncture is a Chinese technique where needles are inserted and they will be stimulating the the large diameter fibers, a particular location, and so that it closes the gate. Acupuncture, acupressure, counter irritants, they all work through this gate control plan. Then there is another thing, that is a transcutaneous electrical neurostimulation. So that means if we can stimulate, because these large diameter fibers, A beta fibers have a low threshold than A delta fibers. We can stimulate the A beta fibers selectively, and when we stimulate A beta fibers, what it happens? It closes the gate because of the activation of the substantia gelatinosa cells. It closes the gate. And this is the purpose. This is what happens in case of the acupuncture, acupressure, counter irritants, and the TANs. Just to revise what I had said about this, the stimulation of a large diameter fibers activate target cell as well as the substantia gelatinosa cells. The target cells transmit the signals to the brain, whatever the signals they are carrying. Substantia gelatinosa cells in turn inhibit the T signaling. So what it does, it inhibits the T signaling, the target or projecting neuron signaling, and close the gate. So this is a closure. The stimulation of the small diameter fibers, these are A delta and C fibers, activate T neurons. Yes, initially they activate T neurons. Then they inhibit the SG neuron. They inhibit the substantia gelatinal neuron. The inhibition of the activation of the T neuron transmit the impulse. The inhibition of the SG neuron, say for example, if this is inhibited and the SG neuron is a inhibitory neuron, so that means inhibition of inhibition, that is what is called a disinhibition. Thus, it opens or keeps the gate open state. In open state, it allows the transmission of the pain to the brain, the continuous. Thus gate is not closed or it remained open. This is about a gate control plan. Now, just to summarize all that, I have made a cartoon here, and in this cartoon, I have made the same concepts of Melzack and Wall, the large diameter fibers and the small diameter fibers. These large diameter fibers are A beta fibers. They carry touch. Thus, and when they come here, they will come to this gate. This is the gate, and there is for this gate there is a SG neuron or a gate person. This gate person is a SG neuron, and when they come here, and they will activate this SG neuron, and SG neuron will say that no, he will close the gate. And if he closes the gate, it will not send the impulses through these target or the projecting neuron to the thalamus and the brain. So they will be closed. So no impulses will reach. Similarly, the pain fibers are carried by the small diameter fibers, A delta and C fibers. When they come here, the SG neurons are inhibited, and if the SG neurons are inhibited, so they will allow the gate to remain open. So that means they will not ask him to close the gate. So the gate will remain open. The impulse is transmitted, and it will reach the brain and via brainstem and thalamus. Now, the brain, thalamus, brainstem, they in turn will have a projection to the SG neuron or the target neuron, which tries to modulate the gate, or either open or close the gate. Here, here we see the number of factors that influence the opening and closing of the gate. So now, what are the conditions which keep the gate open? That means pain traffic continues to reach the brain. So the pain in the lateral spinothalamic tract continue to reach the brain. That is the stimulation of the small diameter fibers, a continuous stimulation of the small diameter fibers, or the negative emotion, the depression, worrying all the time, worrying all the time, or increased activity of the glutamate or substance P, because these are the transmitters. That means if they are not been removed from the synaptic place, then they will activate these C5% and they will continue gate open. So that means, or in the other sense, they are this gate opening. I have already small diameters, A delta fibers. The opiate antagonists, they keep the gate open. Of course, we don't use the opiate antagonist. That means whatever blocks the pain, or the agonists of a substance P and glutamate, they keep the gate open. Now, what are the conditions which keep the gate closed? The stimulation of the large diameter fibers. This is one traffic that closes the gate, so that will not allow the traffic for self as well as to the pain. Then opiates, the, the pain, the morphine-like compounds are enkephalinanergic neurons. So that means the opiates, that means the released, transmitter released from the substantia gelatinosa neuron to this here is enkephalin, or a endogenous morphine. And opiates block, or opiates close the door. That means no pain is transmitted, the person is relieved. The GABA is an inhibitory transmitter that again closes the gate. The 5-HT norepinephrine, if they will close the gate, because they close the gate because they stimulate the opiate neurons, or the enkephalinated neurons, or substantia gelatina neurons, to close the gate. The positive emotions, so that means from the dissenting influences, they close the gate. The social aspects. Here is your party animal. I just mentioned about party animal. The person is social, trying to contact and talk to everyone, and enjoys music, enjoying party. All these activities, these are positive. So that means he will not feel the pain. So that means this is closed. And if one is stressed up, as it happens in case of a examination or any other thing, during stress, a molecule that is a pre-pro-opiomelanocortin is released, and this pre-pro-opiomelanocortin is a compound, large compound with the ACTH or corticotropin as one of the ingredient. Then in addition, it has the opiate. enkephalines or endorphins there, and that would decrease the pain. Or in the other way, if there is a stimulation of the brain stem, especially the periaqueductal gray, that would enhance the opiate or encephalinergic activity in here. So that is how the gate remains closed, the pain is relieved. The gate remains closed means no pain in the traffic. So closed gate is by the opiates, the GABA agonists, the substance P antagonists, the glutamatergic antagonists, the 5-HTergic neurons, norepinephrinergic neurons, or by the stimulation of the large diameter fibers, as it happens in case of a transcutaneous stimulation, or by the acupuncture, or by the stimulation of the brain stem, that is mesenchymal periaqueductal gray. So that would decrease, because that would either through the serotonergic or a norepinephrinergic pathway, that would increase the opiate activity and close the gate. In brief, I have given the gate control theory in this lecture.